Hey everyone. In this video, we will turn our attention to 3 human genetic disorders caused by aneuploidy, all of which are viable. The first is Turner syndrome. Most cases of Turner syndrome are caused by a monosomy of the X chromosome. These individuals are female because they lack the Y chromosome and its male development genes. But they only have one X chromosome instead of the normal 2 for females. This is described as XO, the O standing for the absent X chromosome. This causes various developmental abnormalities, including underdeveloped secondary sex characteristics and short stature. These females are also usually infertile, though conception may be possible with in vitro fertilization or other fertility treatments. You might expect monosomy X to be lethal, since X is a pretty large chromosome with many genes. So why is monosomy X viable? Consider the sex chromosomes of Turner's females, normal females, and normal males. Males are viable with one X chromosome, so it seems only one X chromosome is needed for viability, and in males, normal development. But why is that? The X chromosome has around 2,000 genes. Wouldn't reducing the gene dosage of these genes by half be lethal, as we see for autosomes? It is not. This is because in males and females, normal human development requires that exactly one and only one copy of most X chromosome genes be expressed. In other words, for most X chromosome genes, one copy and only one copy produces the right amount of protein. I'll call this level of protein production 0.5x, which for most X chromosome genes is what Turner's females have. They have the same gene dosage as normal females. Females have 2 X chromosomes, so they inactivate one of the X chromosomes to achieve a gene dosage of 0.5x for these genes, the same as males have. So females have only one X chromosome that's actually expressed. But this leads to another question. Turner's females also have one active X chromosome. So why do they have abnormal development and infertility? Well, I haven't quite told you the whole story about X inactivation. In normal females, not all genes on the X get inactivated. Some genes on either end of the chromosome remain active, resulting in a 1x protein production level or gene expression level for these genes. Since a second copy of these genes are not present in Turner's females, these genes have a lower gene dosage and a lower expression level of 0.5x in Turner's females, which probably contributes to the symptoms of Turner syndrome. In the early embryo, in normal females, the inactivation of the second X chromosome also doesn't occur until the embryo has about 100 cells. So in the early embryo, all genes on both X chromosomes are expressed. The absence of the second X chromosome and the lowered gene dosage in early embryos, 0.5x in Turner's embryos versus 1x in normal female embryos, might also contribute to their symptoms. We'll leave our discussion of X inactivation there because we'll have a lot more to say about it later on in the course. A second form of Turner syndrome is a case of mosaicism, where some cells have 2 X chromosomes while others have only one. As we saw before, this is caused by a somatic mutation that occurs after fertilization and formation of the zygote. When the zygote divides by mitosis, the X chromosomes could be distributed unevenly to the daughter cells. In this example, this third X chromosome may be lost, resulting in one cell with one X chromosome and one cell with two X chromosomes. When both continue to divide, they can produce a mosaic individual. This can produce a milder form of Turner syndrome, as not all cells are monosomic. If some cells retain the third X chromosome, some of the individual cells may even be trisomic, though that is not shown here. A third form of Turner syndrome can occur when part of one of the X chromosomes in a female has been deleted. This results in a reduced gene dosage for only some of the genes on the X chromosome, causing symptoms of varying severity, depending on where the deletion occurs and how large it is. So, to sum up, there are actually three known causes of Turner syndrome. One from a germline mutation, where all cells are monosomy X, one from an early somatic mutation that results in mosaicism for monosomy X, and one that is not caused by monosomy at all, but rather a deletion of part of the X chromosome. A second human aneuploidy is Klinefelter syndrome, caused by an individual having two X chromosomes and a Y chromosome. These individuals are therefore 47XXY, though some cases are 48XXYY or 48XXXY. Because these individuals have a Y chromosome, they are male. However, they have lower testosterone levels, which causes the presence of female-like phenotypes, including narrow shoulders and less body hair. These males are also infertile. Symptoms deriving from low testosterone can be treated with hormone therapy. Symptoms are thought to be caused by higher than normal expression of genes on the X chromosome. Let's see why this might be. In Klinefelter's males, the second X chromosome is generally inactivated, just as it is in normal females. Unaffected males only have one X, so their sole X chromosome remains active. Unlike normal males, which have 0.5x gene expression for all of their X chromosome genes, Klinefelter's males have 1x gene expression for these genes on the X chromosome that escape X inactivation, just like normal females do. This female-like gene dosage of X chromosome genes may account for the female-like phenotypes of Klinefelter's individuals. Some cases of Klinefelter's are due to mosaicism or X chromosome rearrangement, as we saw with Turner's, and these individuals may show milder symptoms. As we said, these males are infertile, so this aneuploidy is not heritable. So if it isn't heritable, how does it occur? At this point, try to answer the question, what are two ways a zygote can inherit two X chromosomes and a Y chromosome? As usual, I'll give you a moment to pause the video. Okay, one possibility is that non-disjunction could cause the female parent to give both of her X chromosomes to her egg, which would fuse with a normal sperm containing one Y chromosome, yielding a zygote with two X's and one Y, conferring Klinefelter's. Second, a normal egg and a sperm which, through non-disjunction during meiosis, gets the male's X and Y chromosomes, could fuse and produce an XXY zygote. Third, we'll consider Down syndrome. There are three types: primary Down syndrome caused by trisomy 21, mosaic Down syndrome, and familial Down syndrome. Symptoms of Down syndrome include various developmental delays and intellectual disability and characteristic facial features. It's one of a very few autosomal aneuploidies that is viable past childhood. Most cases are primary Down syndrome, in which all cells of an individual are trisomic for chromosome 21. This karyogram is of a male, but females can be affected as well. In mosaic Down syndrome, the affected individual is a genetic mosaic comprised of some cells with the normal 2 copies of chromosome 21 and some cells with 3 copies. As we saw with Turner and Klinefelter syndromes, the presence of these normal cells can sometimes lead to milder symptoms. Familial Down syndrome is actually not caused by a trisomy of chromosome 21 at all. It's caused by a chromosomal rearrangement, specifically a translocation between chromosomes 21 and 14, or sometimes between chromosomes 21 and another chromosome type. Let's examine how this occurs, starting with 2 normal copies of chromosome 14 and 2 normal copies of chromosome 21, both acrocentric chromosomes. The translocation is Robertsonian. Yielding a large chromosome that has most of the genes of both chromosomes, and a smaller chromosome that is lost. Only one translocation occurs in this case, so this individual is now heterozygous for this translocation. So the cell now has a large hybrid chromosome, a normal chromosome 14, and a normal chromosome 21. If this cell completes meiosis, there are 6 possible ways that these chromosomes could be sorted into gametes, and assuming independent assortment holds true, all 6 ways are equally likely. Since there are 3 of these chromosomes, a gamete could get 1 or 2 of them. One gamete could get a normal 14 and a normal 21. One could get just the hybrid chromosome. One could get just a chromosome 14. One could get a 21 and a hybrid. One could get just a 21, and one could get a 14 and a hybrid. If this first gamete fuses with a normal gamete, a normal zygote will result. If this one does, the offspring will be what we call a carrier of the 14-21 translocation. These individuals are generally phenotypically normal because they have 2 copies of most of the genes from both chromosomes. If this gamete fuses with a normal gamete, the offspring will have monosomy 21, which in humans is lethal. If this gamete fuses with a normal gamete, it will result in trisomy 21 and Down syndrome. Notice there are 3 copies of chromosome 21 here. It's just that one of them is fused to chromosome 14. But the gene dosage for most of the chromosome 21 genes is still 1.5x, enough to cause Down syndrome. If this gamete fuses, we get an offspring with monosomy 14, which is lethal. If this gamete fuses with a normal gamete, the offspring is trisomy 14, which is also lethal. So out of the outcomes that produce live births, we would expect that 1/3 are normal, 1/3 are translocation carriers, and 1/3 will have Down syndrome. However, in reality, zygotes and embryos that have trisomy 21 spontaneously abort more often, so the probability of having a trisomic offspring is probably a bit lower than 1/3. Notice that the carrier offspring is the same as what we started with at the top, here. The carrier of the translocation can be fertile. They have an increased chance of producing a gamete with 2 copies of chromosome 21, and therefore an increased chance of having an offspring that is affected by this form of Down syndrome. In other words, if an individual has Down syndrome that is caused by the translocation, that translocation could have been inherited from one of the parents, which is why this is called familial Down syndrome. This rare form of Down syndrome can be passed from generation to generation within a family. Okay, so that's it for aneuploidy. In the next video, we'll examine polyploidy. See you all then.